Right, whistle-stop tour of RNA modifications then, in particular the epitranscriptome. So what is the epitranscriptome? Well, it's a bastardization of Latin and Greek which should upset anyone that considers themselves a civilized human, but ignoring that for a minute, it's involving these crazy modifications that can occur to nucleotides that are found on RNA, so that genetic alphabet. It's been expanded by some crazy modifications in some cases, such as that one. Anyway, this is a really cool database. Go and have a look. There's a bunch of them. Over 150 modifications can be found on RNA. However, typically, the epitranscriptome is talking about messenger RNA, which carries the instructions for proteins. Those can be modified, so that alphabet can have little methyl marks or other modifications added on. It doesn't affect the protein that's actually made, but it affects the proteins that that messenger RNA molecule will interact with. So it's essentially a way that RNA is tagged to interact differently with other bits of the cell, so it affects the when and where of protein expression rather than the protein itself, typically. So how did this all come about? Well, this is a pretty cool article. Go and have a read. It kind of starts in 2008, which is perhaps a bit late since a lot of these modifications were actually identified in the 70s, but really it was technological availability and some interesting biological insights. So 2008, it was discovered that if you played with these enzymes that added the modifications in plants, then the plants can no longer grow properly. And then a bit later, it was found that these modifications could be erased dynamically, so they didn't just appear and disappear with the RNA molecule, but they could be added and removed during the RNA life cycle. That was the suggestion. The fact that it's dynamic is really exciting because it might mean that it's druggable. And then some other techniques came out that meant you could map and find out where and when these modifications were added to RNA. These techniques are still relatively limited, but they're rapidly evolving now. This is a new field, it's only 10 years old. And now the epitranscriptome is an exciting part of the biotechnology revolution that's currently happening. And biotechnology in itself is worth around 2 trillion across the EU market. So this is only a small piece of the pie so far, but I'm expecting that this is going to grow quite a bit over the next few years. Great article, go and read them. Start with Medomics, have a play, figure out what modifications happen where, go and have a look at the epitranscriptome on Wikipedia, and then go and have a look at this cool article that just sort of gives a little bit of the background of some of the groups that have been working on it. Wicked. Right, bye.